The central venous access device is an essential tool for our critically ill patients in ICU. And unfortunately, they're really vulnerable to mechanical and infectious complications. They're really vulnerable in the IJ. And in ICU, you have a lot of administration sets that are attached to that device, and it's really heavy. So, these dressings are fighting against gravity and against, forces that are associated with those lines. So we did a study back in 2016 where we looked at different ways to secure those particular devices, because we'd found that there was high failure of those dressings. And if a dressing's lifting, it means that there's that chance for organisms to enter what we call it's a wound, and that can lead to a local or a bloodstream infection. So we wanted to look at how we could best secure those devices. Mastisol is a non-water soluble gum mastic. The purpose of it is to help dressing adhesion. So we took this home with us as an idea and developed a protocol to be able to look at this product with our jugular lines. We're looking at standard care compared to standard care with Mastisol liquid adhesive. We really wanted to stop those lifting dressing edges that we'd seen in our previous research. But we had secondary outcomes we were interested in as well. So we wanted to look at, if we had MLA compared to standard care, how long were our dressings dwelling for? We wanted to look at how many dressings we needed to complete treatment. These are really important from a health economic point of view. We wanted to look at whether those dressings were failing but not being replaced. Because it's pragmatic and things happen in clinical practice, we wanted to understand that. And because we had never used this product before, we wanted to really understand risk profiles associated. We wanted to know what was happening to the skin. Were there medical adhesive related skin injuries associated with the product? And were staff happy with the product and patients? We wanted to have patients that were going to have the line in place for at least 3 days. That was important. We wanted to make sure that we could look at those outcomes over time. We would try to make sure that we always recruited people that were going to be in ICU for greater than 24 hours. And we also wanted to get them within 12 hours of having that device inserted. We had exclusions for these patients, and they included patients that already had an active bloodstream infection, and that's because a really important outcome is looking at infection, and we didn't want there to be any confusion as to where the source of that infection was. And it was really to adhere with the CDC guidelines around definitions of infection. In our MLA group, the dressing failure was 28%, which was almost half what we saw in our standard care at 50%. And this was a statistically significant result favouring MLA. We also had a look at how long our dressings were actually dwelling for. And in the MLA group, on average, the dressing dwell was 58.5 hours. And this was almost twice as long as what we were seeing in the standard care group at 23 hours, which was a significant result favouring the MLA. We also evaluated the number of dressing changes that happened, so the number of dressing products that we used, and it was 64 in the MLA group and twice as high in the standard care group, with over 120 dressings being used during the course of time that device was in place. Just because a dressing was starting to lift at the edges didn't mean that the clinicians immediately replaced it. So we did daily site inspections of the different dressings, and one of our measures was to look at how many dressing edges were lifting. So the median score for this in our MLA group was 0, whereas in the standard care group on daily inspection, we found that there was at least 1 dressing edge lifting, and this was also a significant result that was favouring the MLA. We looked at patient and staff satisfaction, and they were satisfied with this product. It didn't cause our patients any at removal they were just as satisfied as they were with our control product, so that this was of benefit for us as researchers and clinicians to look at introducing this product into our practice. We did a sub-study of 20 patients. This was a convenient sample. So the reason it had to be convenient is because the research nurse had to be present at the time a dressing change was happening in the intensive care. So what they did was take a skin swab at dressing change, because we wanted to look at the amount of microorganisms that were on the skin. And what we found is that we had 12 positive growths from these specimens. We had 8 in the standard care group and half as many in in the MLA group, which is potentially because the dressing is adhering to the skin better, and we're not getting that tracking of microorganisms to across the surface of the skin and potentially to the insertion site, which can lead to local or systemic infections. We recommend that if you are an ICU and you are, using a jugular central venous access device, that we think that there is benefit to using this product. So we would recommend other ICUs to adopt this, and hopefully they'll have the same benefits as what we've had.